Yes, good evening to all the participants. Welcome to the day 5 session, day 4 session on virtual screening methods. So, today we'll be discussing on virtual screening, which is, of course, a virtual screening based talking. But before going to this topic, can we recap the last session topics? What exactly did we discuss in the last session? So kindly unmute and answer, or you can type the same in the chat box. Can I ask you specifically, like what are the tools and databases we have discussed for determining ADMET properties? For determining ADMET properties. So come with few examples. I have provided so many demos, isn't it? I have showed you around 4 to 5 tools for ADMET, including databases.
Sir, Swiss ADME,
Uh,
Pro
uh,
ADME.
Madam, okay. So your voice is actually breaking often. Uh, can the participants type in the chat box? Drug bank. In drug bank, we can find the ADMET properties. You're right. So that is the database for finding the ADMET properties. Binding TB, you can get the information. Good. Swiss ADME is the tool. Okay, whatever Kalaivani has mentioned is related to databases, and Yashwant mentioned is related to tools. ADMET and Protox is for determining toxicity. Of course, it's a tool. But CHEMBL and CHEABA are databases. Here we can determine only physicochemical properties, not the ADMET. Physicochemical properties, I mean, what is the molecular weight, number of hydrogen bond donors, acceptors, like that. However, you find in NCP PubChem, similar kind of data you find that. But other than Swiss ADME, Protox for toxicity, what else did we discuss? You remember, I told you pre-ADMET and PKCSM at ADME. So these are the tools, and also click to drug. Okay, so this is another tool for determining ADMET properties. So other than Swiss ADME, you can use all these tools. And databases generally we can find the physicochemical properties, not exactly pharmacokinetic properties. Okay, I think it's a kind of a twisting question, but many participants have interacted. I'm very happy that many participants are giving somewhat relevant answers, and more few participants given more accurate answers. Now let me come across the next question. Uh, in the next question is, what do you mean by L in LADMET? What is L? So before that, very good. The answer is liberation. Good. See, even one new participant is answering this. So you're listening very carefully. Thank you. Good interaction. And what else did we discuss? We discussed about protoxicity, about various toxicity. You remember? I think you can practice all these things. Everyone can able to practice because it's a free online and offline tools. And now let me go ahead with the today's session. We have plenty of things to learn in today's session. So I'll be teaching you hands-on session in Autotalk PVRX. So let me go ahead with virtual screening methods. So today we'll be discussing on what is virtual screening and a method of virtual screening, that is lead hopping, high throughput screening. Okay, and we can also see few more. This is another mode of classification of visual screening methods. We can classify as a ligand based. This is one number one, structure based and hybrid based. This is another mode. And visual screening softwares, you can see these tools we can use as a screening of drugs. For screening multiple drugs, okay, to a single drug or a few important drugs, we can use these tools. and we'll be discussing on how to practice the Autodoc PYRX. So related to drug discovery. In drug discovery, we initially take the first step to identify the target disease. And after identifying the target disease, for which disease you are coming up with a drug, then you take a next step in a pipeline to find what are the metabolic pathways you can find in the disease. If you find nearly 8 pathways and only 7 pathways are explored, the one pathway which is not explored, that you can take as a problem statement and it can work. In that metabolic pathway, even if they have worked, but one target protein they didn't focus, then you take that protein. That is step number 3. Whatever protein is not taken in interest, that we can work in the form of a project. Okay. Now, for a target protein, what we anticipate is the 3D structure. Okay. Later, however, we find a protein preparation. The next thing, okay, this is something related to protein preparation. For protein preparation, we are taking these 3 steps. The next is ligand preparation. For ligand, we are not going to take a single ligand, but we take multiple drugs, multiple ligands. So initially, we have to screen. For screening the virtual library of ligands, we can use the drug likeness. Okay, drug likeness is one of the filter virtual screening method, and number two is the using the ADMET properties. So ADME you can use separately, toxicity you can use separately, and then mutagenicity, carcinogenicity. You have different tools to determine. Just upload a SMILES format. You find all this information, and then we can come across with pharmacophore based docking or pharmacophore mapping. Then we can go with the virtual screening based docking. Virtual screening based docking. So, and then we can take further lead hopping, other types of virtual screening methods, which I'm going to teach you in the next slides. High throughput screening, lead hopping, etc. Then we will take finally MD simulation. MD simulation. So this is how we generally connect. Actually during the docking, we need the 3D structure of ligand, and we need the 3D structure of protein. So these two we try to dock. That is a kind of a screening, and from that screening we identify the leads. Okay, again we can go with lead optimization. So this is how the pipeline is used. This is one mode of classification of virtual screening. That is a ligand based methods. You can find similarity search, pharmacophore mapping. So tomorrow we'll be discussing more about similarity search. So this similarity searching comes under chemoinformatics. Chemoinformatics. Okay, so we'll be discussing more about that and pharmacophore mapping in subsequent sessions. And today we'll be discussing something related to virtual screening based docking. Okay, that is coming under structure based methods. So this is one classification, one more mode of classification. That is how the ligand based screening or broadly classified. Okay, so you can find in literature papers and journal papers, but whatever we want to learn in practical aspect, that I can focus now. So if you see in virtual screening, okay, if we have the first method, a default virtual screening. So here we screen multiple ligands. Like if you have multiple ligands in a single data set, I told you in drug discovery hackathon competition, which has happened in 2020, I have taken around 1,500 compounds from PubChem, okay, in the form of a data set, and try to talk with spike glycoprotein. Remember that kind of an example, whatever I mentioned. To do that, initially, this is one of the most important filter I use, that is virtual screening based docking. If you do virtual screening based docking using a tool called P5RX, Python RX programming. So this is Python RX programming is a product of actually MGL tools Autodoc. In Autodoc, we have different types. One such type is Autodoc PYRX. This is used to dock multiple ligands simultaneously with a single target receptor. Okay, so this kind of virtual screening, if you see, the accuracy will be somewhat low. But if you talk a single ligand with a single receptor using Autodoc, the same Autodoc, Vina, Autodoc 4.0, covalent Autodoc. So in all these types of Autodoc, you can able to see that the precision will be more. I mean, the accuracy is more. Okay, but here if you try to talk with multiple ligands, accuracy will be less. But you know the time taken is very less if you go with Autodoc PVRX. Maybe 1 in 20th. Okay, if you take 20 minutes, you can able to complete in 1 minute, like that. Okay, so you can able to do as soon as possible with Autodoc PVRX, that too with multiple ligands. So it's a boon to complete even initially doing a virtual screening using this Autodoc PVRX. Then how you validate the results? So whatever we talk, multiple ligands can be, this is ligand 1, ligand 2, ligand 3, it goes up to the ligand N. So how many ligands we try to talk with the protein target? Okay, for all these ligands, we generate the results in binding energy. Whatever binding energy we generate, it will be in kilocalorie per mole. This is the unit. The lower the energy, the highest stability, highest binding affinity. This is how we can conclude the lowest binding energy. So the negative maximum is what we prefer. For example, if L1, I get a score like minus 9.2 kilocalorie per mole, and for L2, I am getting minus 7, which is negative maximum. From the results, it is evident that L1 is negative maximum. So this is the best one when compared to ligand 2. So this is how we screen the compounds through binding energy. Of course, for calculating the binding energy, we use the change in Gibbs-free energy calculation. Whatever we learned in thermodynamics, del G equals del H minus T del S. You remember this kind of a formula they have used. Okay, now you understand what is virtual screening. Now let me go ahead with a type of virtual screening called lead hopping. What is lead hopping? The lead hopping is also called as fragment based docking screening. Fragment based docking. You know, actually, what is happening in this lead hopping or fragment based docking? Initially, we take a lead, initial lead. So this is the structure of the initial lead. The drug, whatever, is already used for treatment of one particular virus. But if I want to use the same drug for another virus, whether if it is not giving a good binding affinity, then what we can do, we can fragment. So we can fragment here. So that's what happened here now. Whatever we fragmented, we try to attach in another functional group, which is closer to this. In the double bond, we are attaching with a linker. Whatever I am using in this yellow color, now I am going to link a fragment 1 and fragment 2. This is fragment 1, this is fragment 2 with a linker, which is shown in yellow color. So this is what we call it as fragment based. Now, after fixing this kind of a new lead, whatever we generated, we try to talk with the same target, like a new virus, whatever we want to target. And if that this new lead is going to talk with minimum binding energy, then this is best over when compared over initial lead. This is how we can come up with new leads for new type of viruses or any kind of new diseases. Okay, any diseases, we do this fragment based approach. If you combine with talking, we call it as fragment based talking approach. Now let me come across the third step, that is increasing high throughput screening hit rates. Here you can see multiple compounds are available. We try to talk, we we try to talk, okay, with a same target receptor, then how it varies with the normal virtual screening. For this, I am going to explain with an example. See, this is a project I guided in computational part. That is a lactose shield project done by SVCE 2016 IGM team. IGM is International Genetically Engineered Machines. So in this team, they have secured, I believe, silver medal. Okay, so for how they have done. So the students from SVC have gone to Masters Institute of Technology and presented this. So how they did it, actually, the main theme of this concept of this is to protect the milk. So can someone tell me what is the lifespan of the milk what we procure? Can be Avin in Tamil Nadu, Nandini in Karnataka, or Orokia, whatever is available in your place. What is the lifespan of milk? So you can unmute and answer for this. I'm just asking a general question, but that is something linked with competition part.
So boiled milk we can have 1 day, sir.
One day, even 1 to 2 days. Then what else, ma'am? If we store in fridge, we can have around the same or even 2 to 3 days. Okay, so this is how we can extend, isn't it? Not more than 1 to 3 days. If you keep it in fridge, we keep it in 4 degrees centigrades.
Maybe in freezer 4 days, sir, maximum.
Yeah, in freezer minus 20 degrees centigrades, the milk won't be getting spoiled. Even we can store up to a week, not more than that. But this is how. Okay, so you mentioned like up to 4 days. But actually, there is one type of milk called ultra packed milk. Have you heard about this ultra packed milk? So this kind of a concept I heard only during the pandemic time. There are some milk packets which are nearly 3 times expensive when compared to the normal milk. The futures of this ultra packed milk is we can able to store the milk for 3 to 4 months, not 3 to 4 days, 3 to 4 months. At that time, we find the dairy products are very deficient during the pandemic time. So during that time, they did this. This concept is similar to the ultra packed milk. Why the milk is getting spoiled, like whatever mentioned, the milk span is coming around 1 to 2 days. What is the reason? The reason because of the microorganisms. The microorganisms can spoil the milk. So to get avoid the spoilage, okay, so we have designed a project according to the concept the previous literatures. We came to know that if we use a peptide, okay, of 9 amino acid peptide, okay, the last three letters of the peptide should be tryptophan, and we should have nearly six more amino acids have to be filled. The six more amino acids have to be filled. You know that we have 28 amino acids. So under this case, okay, this first amino acid can be any of the 20 amino acids, including W. Like this, you can have for the other other five places. Then you have a combinations of peptides. Thousands of peptides are formed if you have this kind of a strategy. Okay, according to the literature, we should have the last three, that is the tail of the peptide should be the tryptophan of 3 amino acid residues. Then uh, we have synthesized these kind of a compounds using the combinatorial chemistry, that is through the computational techniques we have generated the compounds, I mean the peptides of 9 amino acid, and whatever 9 amino acid peptides combination, we try to talk with that microbes. The microbes genes which can spoil the bacteria, I mean which spoil the milk. Got it? The bacteria genes, some genes are responsible for spoiling the milk. If you want to target that kind of a genes, that kind of a receptors in the bacteria, milk spoiling bacteria, we have to talk. through the dock with the receptor. So this kind of a project, you know, we worked and we come up with a kind of results, and the students secured silver medal. I just guided only the competition part. Now, let me come across with this kind of virtual screening. I told you we have ligand-based, structure-based, and also we have hybrid-based. The hybrid-based is in between the ligand-based and structure-based. Okay, so this is the example. Now let me come across the two softwares. The softwares we can see Pharmagist, PYRX, Ligand Scout, and Doris. Today we can see at least one tool how to practice. The tool what I'm going to demo, provide you the demo is Autodoc PYRX, Python RX programming. So the source, if you want to download this tool, we have to download only one software that we can download from this URL. The URL, whatever I typed in the uh, Google Meet chat box, you can see. If you click that link, you can able to download one exe file which is compatible for Windows. Initially, if you want to dock a single, I mean multiple ligands with a receptor, we have to download multiple ligands, isn't it? Whatever multiple ligands you download, that should be in a single file, dataset file. For this, you can use any chemical compound databases. Can be ChemSpider, can be Drug Bank, CHEMBL, any databases. Okay. Myself, I have used NCBA PubChem. I'm going to teach you with NCBA PubChem. Okay. There is also one more database called Human Metabolite Database. Here you can find all the metabolites, okay, of plants and humans related to. Now let me go ahead with how to practice with Autodoc PYRX. Okay, so even if you want to download the kind of download the data set, you can also download the data set from Enamine. Enamine is a compound library. So here you can able to see we have different data sets. Essential fragment library, we have some compounds. High fidelity fragment is 1920. So disease specificity index watched library 860 compounds. Like this, we have different libraries. If you click this kind of a link, okay, you can download. So you find the links for each and every libraries. Now for installation, obviously we have to use pyrx.sourceforge.net. Okay, and then how to work with it, that I'll be explaining you step by step. Now let me go directly to the web resource. If you want to download, so what is the URL we have seen here? I just click that link, it directly take you to that. So now whatever I'm showing you is the source to download the PVRX. So if you download the PVRX, the software of PYRX. I'm checking whether we have that kind of a file somewhere here. Yeah, then I can directly take you to that browser if not available. Okay, let me go ahead with this. Yeah, so there is a free version. So if you click this free version, if you click this, you can download the Autodoc PYRX. Okay, we also have commercial tools, but the free version is sufficient. Anyhow, today I'll be sharing you the link. So after downloading and installing the PYRX, this is the icon you can find. So the icon will be like this, like Aladdin lamp. So if you want to open this kind of thing, simply double click. Actually, Autodoc tools are commercial softwares, but we can skip the registration, the license registration, and we can work with it. So there is some problem with, yeah, not so. So presently, we have to download the dataset file of chemical compound. For chemical compounds, I'm going to take NCPA PubChem. So in NCPA PubChem, I'm going to take a group of compounds. So for a group of compounds, I'm going to try with this query we know. Poirevin is a rotinoid from Poirevia diffusa. There is a plant called Punarnava. Punarnava, like for blood rejuvenation, we can use this, especially for treatment of kidney stones, treatment of urinary infection. Okay, if you use that Poirevia diffusa, in Tamil we call it as Mukaratai Tirai. You know, in this Poirevinones, we have 15 types of Poirevinones. When I searched, I found 15 compounds. The compounds will be like Poirevinone B, Poirevinone E, like you have A, B, C, D, E, F, G, something like that, up to 15 compounds. I want to check which is the best compound for a particular target. So if I want to download these compounds, but I don't want all the 15 compounds, if I want to screen few compounds from these 15, I can able to screen the compounds by clicking refine or filter. If I click refine or filter, it takes you to this page. Okay, in this page, you can use the filter for physicochemical properties. Like for example, the donor count, I'm going to keep like 2 to 3. Okay, so this is one such filter I'm going to keep at present. And now I'm going to search. I'm checking only two compounds are filtered. I mean, out of 15, only two are eliminated. We have only 13 compounds at present. And if you want to further screen, okay, with respect to hydrogen bond acceptor count, I'm going to give like 5 to 6. Or even it can give whatever properties you want. Okay, so based on a literature evidence, you can able to do the filter. Now, whatever I've done the filter, I zoom in and show it to you at present. Now I got only 5 results. Initially, I had 15 compounds, but based on these filters, I filtered to 5 compounds. Now, our objective is to download this file. So, to download this file, click this download and don't use coordinate 2D, which is present in default. Go to 3D. Okay, and the compression should be none. No need to go for a zip file. So, after changing to 3D, this alone I have changed and click this SDF, structured data format. So now I'm going to download this SDF. So from beginning, I'm using CADVD 26 folder. I'm going to download this. And now I'm going to save this compound dataset as Poirevin nodes. Poirevin nodes.sdf will be in default. Okay. So if I want to open this dataset, so double click and see the compounds, how it looks like in Discovery Studio. So Discovery Studio, I believe you have installed. This is most important. Without this, it's difficult to learn up to the final thing. See, I got only 4 compounds, but when I don't before downloading, we can able to see 5 compounds. Why I got 4 compounds? In 3D we got only 4 compounds. In 2D we have 5 compounds. Okay, that's the reason. Now, if I want to see all these 4 compound structure, select all this. Because default it is in 2D, isn't it? When I change to 3D, whatever 3D structures are available, that only you can able to download. Now, select all these 4 compounds, show in graphics view. So now check these compounds. All these 4 compounds are placed together. You find that based on the functional group, slight changes there, isn't it? So all the 4 compounds are present in this now in a single file. Okay, now we have created a data set. Okay, so the input chemical compound file is created. I've already pasted one protein structure from PDB website, protease. I want to check which is the best compound which is going to talk with this protease protein. So this is the problem statement I'm going to work on at present for a demo. So now I just open this Autodoc PVRX. Okay, after installing that one exe file, you get this kind of a tool. No need to install any other tools. Okay, now the next step is first you have to go to the edit preferences. I'm audible to everyone. Just interact, yes or no.
Yes, sir.
Thank you. Now, thank you. Now let me go ahead with the workspace. In the workspace, Okay, we have to fix which directory we are presently using. Okay, so presently I am using a directory in desktop called CADVD 26. So in desktop, I selected this folder, click OK. So workspace is like that. So I am using i7 laptop. So the CPUs, number of CPUs are more, up to 19. If you use i9, you get few more CPUs. It means that how many threads you are going to use to run this output. So based on the processing power, you can able to complete it as soon as possible. Now let me click OK. Only thing what I changed is I changed the workspace alone to the input file directory. Now click OK. This is first step. The second step, I can't able to zoom in because this is a software, open source software, I download and install. Only thing is that you have to watch even the recorded video if the font is not visible. But I will be explaining you what exactly I'm doing. So first thing, edit preferences. The next step is file, load molecule. In the load molecule, go to the desktop, go to the folder where I downloaded this protein file. The protein file what I'm going to use is a protease. Simply click this protease. So this is the protein structure in wireframe model, which is going to open here. Okay, this is a kind of a Python molecular viewer, which is embedded in this Autodoc PYRX. Okay, now after opening this file load molecule, you can able to see here. So if I expand it, you can find only the amino acids. You don't find anything else. That's it. Okay. After loading, you get only this alone. Okay, protease, and you don't find anything here. The next step, what you're going to do here is simply click here, click this protease, right click, Autodoc. Okay, make macromolecule. After loading, right click, Autodoc, make macromolecule. Do this. Completed. After this step, if you go to the Autodoc, now under macromolecule, you can find once again protease. Earlier we don't have. If I expand it, you find protease.pdbqt. Participants, remember one thing which is most important in Autodoc. Autodoc, we can able to do docking of a single ligand or multiple ligands with a target receptor. In both the cases, it can be any type of Autodoc, Autodoc PINA, Autodoc 4.0, covalent Autodoc, or Autodoc PYRX. We have to convert both the ligand and target receptor files in the form of PDBQT. Earlier, it can be in any formats, .stf, .pdb, .mol2. But our objective is to first initially convert into PDBQT formats. Because Autodoc accepts only PDBQT formats of both ligand and receptor for docking. Okay, so after doing this step, you got this kind of file generator. If only if you click this make macromolecule, then only PDBQT of protein is created. What is the next step? If you have prepared a protein, the next step is to prepare the ligand. To prepare the ligand, go to the Autodoc wizard. No, instead of Autodoc wizard, go to OpenBabel. You know why I'm going to this OpenBabel initially? The OpenBabel is a open source software to convert any formats to the desired formats. Initially, whatever ligands I have downloaded from PubChem are in SDF format. I have downloaded 4 structures, 3D structures of compounds in a single SDF file. Okay, to convert this into PDBQT, I am going to use this OpenBabel. The OpenBabel is an exclusive tool to convert. Even if you go search in Google, you get a OpenBabel, a different link. And in the online, you can either use an online tool or you can use an offline tool called Babelwin to convert any format into any formats. Now let me first do this step. OpenBabel, which is already merged with this Autodoc PVRX tool. The next step is to click this insert new item. See here in the green color icon. When I place my icon, you can find here in insert new items, Okay, you have to go to the desktop, go to the where exactly I've saved the file. The file I saved is verivnons.htf. Select that, click open. Now all the four ligands are shown here. The next step is click one ligand and now right click. So if you use this minimize selector, so do only for one ligand. What is this minimize first of all? So minimize here we mean doing the energy minimization. This is an important protocol we do. Okay, this is an intermediate protocol. Okay, we do to convert the ligand more stable. So initially the energy of ligand is more, but if you do the energy minimization, the energy will be reduced and the energy that ligand will be more stable to the environment. Okay, for this we do the energy minimization. So instead of clicking minimize selector for a single ligand, I'm going to click this minimize all so that the energy minimization can be performed to all the ligands. After clicking minimize all, you can see the energy values are also displayed. Energy minimization is done for all the four ligands. The next step is again click only one ligand, any one ligand, and then convert all Autodock ligand to PDB-QT format. Our purpose is to dock all the four ligands with the receptor. So to convert all the ligands instead of convert selected. Okay, now I'm converting all the four ligands to PDB-QT. How to find whether the ligands are formed in PDB-QT? Double click here. If you double click here in this Autodock, you find all the four ligands in PDB-QT. Before that, we don't have these formats. Now only it is generated. Okay, so this is the step. So the most important step is to convert the ligands and proteins in PDB-QT. Now this is completed. Okay, the next step is go to Vinavisa. After completing the open babel and all these two steps, we have to go with the Vinavisa. In the Vina wizard, it directs you step by step. The first step it's asking you is to click start. So click start. If you click start, you can able to find a window. In this window, you can see the protease.pdbqt is already get selected, but no ligand is selected. Protein is selected, but we have four ligands. The four ligands are not selected. Then we have to select the four ligands. So for this, I left click here and I use shift followed by left click. So all the four ligands selected. If all the four ligands selected, now the data is changed to four ligands selected. And even the protease.pdbqt is already selected. So if only if you select all the ligands and protein, then only can go with the next step for docking. The next step is to click forward. If you click forward before docking, we get one step called grid generation. So grid generation is an intermediate step which is present in all the types of Autodock. Now, of course, not only in Autodock, even if you see any commercial tools like Discovery Studio or Scroid Incher, Cybele, any tools use this grid generation. The purpose of generating the grid is like if you have a ligand and if you want to dock a ligand in a protein, you have to find exactly in which atom of a receptor it's going to bind. In a receptor, in a protein target, what you can find? Amino acids. So plenty of amino acids you can find. In this amino acid, you have plenty of atoms. So whatever ligand we are going to bind is going to dock on which atom of an amino acid in a receptor. You can able to find if you can able to uh, encapsulate the entire protein in a grid box. In a grid box. To encapsulate this protein in the grid box, okay, so we can also use this 3D isometric view. If I use an isometric view, you can able to see a 3D part. See whether the protein is entirely encapsulated, not, isn't it? So simply click here. Now I try to encapsulate the entire protein. Okay, I'm going to cover the entire protein using this. Okay, so now it is our protein is packed inside the crate. You know, this crate you have multiple grid lines that is there in default. Do you know what is grid and grid lines? Like if you want to take it in a graph sheet, okay, for example, if you take a graph sheet, we have a major scale as centimeter scale, isn't it? In y-axis and x-axis, you have a centimeter scale. But we also have minor grid lines, that is scale. In 1 centimeter, you have 10 lines. In a cubical box, you have 10 rows and 10 columns in 1 centimeter box in our normal graph sheet, whatever we use it in observation reports. Similarly, here this grid box is split into multiple grid lines. So even a trace of an atom can be identified. So if a ligand is going to bind with an atom of an amino acid, you can able to find through X, Y, Z directions. For this, we are using this. Now, after doing this step, the next step is to click forward. That's it. We have completed this step. Very few steps. And this is somewhat good when compared to online tools. There are also online tools do the virtual screening, but when compared to that, working with open source software gives reliable results. But if you want to further increase the accuracy, I suggest you to do with manual docking using Autodock. Now we have also generated the results. How much time it has took? Not more than 30 seconds. All the 4 ligands are docked with the protein receptor. Even if I dock 15 ligands, it may take not more than a minute. Okay, now our next objective is to download the results. To download the results, we can click this save as comma separated value files. See here in bottom we have CSV. Click this CSV and go to the desktop wherever folder I just have. I'm going to paste this. So this is actually ORAV nodes docking with a protease. Now I save this. After saving this file, you can see this Excel file. Simply click on open. Okay, so I'm going to use, we're just going to increase the font. Now I'm going to use a filter. So under the filter, data filter, so if I want to screen which is the best ligand docking with a protease protein, a target, a sample target, whatever I have, so simply click here and see here, the base docking energy is minus 8.6. The minus 8.6 whatever we got is for this pubchem ID. So just copy this pubchem ID, okay, whatever we have, and simply go to the pubchem, okay, and then you search. If I search this pubchem ID 14018346, it is for even on A. So out of after screening the from the properties, 4 out of 15 are screened, and out of docking all these 4 compounds, I found variable A is the best compound, which is binding well with the protease target. This is how we have to conclude. Now I just try to remove the filter and I can show you what exactly we have. You know, for each and every ligand, we have generated nearly 9 poses. If you see, there are 9 poses. The first pose or first confirmation, you can see root mean square deviation as 0. It means that the conformers are ranked based on the negative binding energy. Which is best fit that is shown at the top. Okay, the results are shown in descending order according to this. So this is the best conformer for the ligand. This 5324349 PubChem ID ligand. And this is for the second ligand 14018346. That's what they found it as per known A. Okay, we have 9 poses. And this is another ligand, 1 4. So like this, we can able to find 4 ligands, and out of these 4 ligands, no doubt the best one is the pyrimidine A. So this is how we can able to find the binding energy. Okay, so this is how we can able to find the binding energy. Okay, now let me come across the next thing. So we have completed how to talk the Autotalk PVRX and how to infer the results. Do you have any doubts at present in this? Yes or no? If and if you practice, I think even some more doubts will be clarified, isn't it? So I'll be sharing you the exe PVRX exe file as well as the sample files, whatever sample file I used. So I'll be sharing it in the folder. You can try to do with it, and then you can try with your own kind of research samples. I mean, the files downloaded from any of the public domain databases. So as we completed earlier, okay, so now we are going for the next chapter, network pharmacology. So we have 10 more minutes. So for the 10 more minutes, so the virtual screening is completed now because network pharmacology, we have much more thing to learn. So I'm just going to initiate this network pharmacology. So in under this session, we'll be discussing what is network pharmacology, what is system pharmacology, how about pathways networks, what are the types of pathways like interactomes, what do you mean by diseasome, and how to work with network visualizer like cytoscape. And we can also discuss with some case studies. And today, I'll be sharing you few journal papers what I published on computational studies. So whenever you feel time, you can go through that. And I believe few participants joined late in the WhatsApp group. So I'll be sharing you once again with the YouTube compiled videos, that is which I delivered to the top MNC companies of bioinformatic companies. Okay, also watch these videos to get something like how the pipeline is constructed and a basic thing what is happening in the bioinformatics industries, etc. Now let me come across with the related to this network pharmacology. So what is network pharmacology? Network medicine is the application of network science towards identifying, preventing, and treating diseases. Okay, that is, if you want to come up with a kind of a precision medicine, nowadays, many startups are getting flourished and getting much of grants. The grants for developing the precision medicine ranges from 1.5 crores to 25 crores. This year, it is up to 25 crores. In next year, it can increase even, it can scale up even up to 50 crore funding. So we have plenty of opportunities in developing the precision medicine. What do you mean by the precision medicine? Tailoring the medicine according to a specific patients. We can't able to give medicine like acetaminophen for treating common fever for all the patients. For most of the diseases, can be breast cancer if you take. Okay, even the survival rate is around 63 percentage, but even we can find the fatality more than 30 percentage in 5 year survival. Okay, and if you take this pancreatic cancer, kidney cancer, survival rate is 5 year survival rate is around 10 percentage. It varies with respect to the regions, especially based on the alleles. Okay, the allele of patients or somewhat few alleles of patients are immune to few cancers. For example, if you take US Americans, okay, they are more prone to get melanoma. Melanoma is nothing but skin cancer. Okay, they are more prone to get skin cancer. Okay, so more than 2 percentage. That is, out of 100 people, people will be getting melanoma. But if you take Indians, it is 0.00005 something like that. It goes with many decimals. Very rarely. Okay, so that kind of the cancer is occurring just because of the genes, the alleles. Okay, HLA alleles. The Native American will get like this. But if you see the Indian American, so the Indian American one day I'm telling 0.0000005 something like that. Indians very rarely get this melanoma. So it varies with respect to alleles. So if you are focusing on developing drugs or vaccines, focusing and targeting on alleles are also important. Alleles, again, once again, I'm telling HLA, human leukocyte antigen alleles. So this varies with respect to person, like how each and every person varies with respect to fingerprint. The faculty members are doing the fingerprint for uh, capturing the attendance, isn't it? So you can know that even the siblings can have vary with respect to fingerprints. But I came to know that a few siblings have also have same fingerprint. I mean, same kind of fingerprint. But the HLA fingerprints vary even to that kind of twins. HLA fingerprints definitely vary even if they're born as twins. So the drugs or vaccines has to be varied accordingly. For this, we need this network pharmacology approach. Just targeting multiple drugs with a single target or multiple drugs target or a single drug targeting multiple targets. This is the basic idea in drug design. But what exactly is happening at present is the development of precision medicine. So for this, one should know PPI, protein-protein interactions. Okay, then biological networks, the most important is protein-protein. And we should also know the metabolites. Even the metabolites produced, okay, can also can influence on treatment of diseases. So everything is important in the treatment of disease. And how about the recombinant protein production? So even in the recombinant production, the metabolites produced during the synthesis is of course most important. Okay, so it can be recombinant kind of metabolites production, or even it can be working on disease in the. So we need this information. PPI, gene regulation, gene interaction. These are important. There is something other than PPI, other than gene interaction, gene regulation. We also have something called disease networks. In disease networks, we can come to know that one target can cause multiple diseases. For example, if you see EGFR, epidermal growth factor receptor, this is a common target for almost most of the types of cancer. For ovarian cancer, cervical cancer, breast cancer, all the types of cancer, I find this is of course one of the most important target. So one target protein responsible for multiple cancers. So then how about that? Okay, how we can treat exactly breast cancer instead of cervical cancer? So more complexity is there, isn't it? For this, we need these kind of disease networks, which map relationship between diseases and biological factors. One thing. Before knowing one thing, I just want a kind of interaction from your side. Can someone tell me examples for autoimmune diseases? Autoimmune diseases. Any examples? Just give me some 2 or 3 minutes to complete this session. What are the examples of autoimmune diseases? You don't know one, any one. Cancer, we can take any organ and we can tell as a cancer. Mouth cancer, tooth cancer, tongue cancer. We have all the types of cancer in mouth itself. But how about autoimmune disease? Give me some few examples. Also, what is that? Can I give some examples? Psoriasis.
Type 1
Ah,
diabetes.
very good. T1DM. Okay, so we have given one more example, T1DM. See, T1DM is something different. Just because of insulin secretion, insulin resistance, we find these kind of a disease. Asthma, this is a respiratory disease. Psoriasis, this phenotype is entirely different. That is, it's related to skin disease. Skin disease, respiratory disease, metabolic kind of a disease. Okay, there's linked with insulin resistance. If you find this kind of diseases, all these diseases, if you see the most important genes, if you find 100 important genes for these diseases, so you can find only one or two genes alone or different. That is, 98 genes are responsible for causing, the 98 top genes are responsible for causing any of these diseases. But one or two genes distinguish the type of diseases. You got it? What exactly I mean? In autoimmune diseases, the genes causing the autoimmune diseases are almost same for even these types of diseases. So maybe 95 genes or 98 genes based on the type of autoimmune disease, you can find the same common genes are responsible for causing this kind of disease, but only some 4 or 5 genes will be distinguished. So that is a pathetic situation. Okay, there is much more thing interesting to learn in the network medicine. Still, we have 2 more minutes. So let me explain few more interesting facts. This is something theoretical. I'm not explaining practical. However, we'll be discussing more practical on tomorrow in this network medicine pharmacology. So one more example I'm going to provide is Zolgensma. It is more, the expensive medicine in the world. You know, the Zolgensma is a gene delivery based kind of a medicine. So just now I was mentioning about the genes. You remember? I told you based on genes only we get we have to come up with a kind of a precision medicine. You know this Zolgensma, which is a brand name, uh, which is produced by the Novartis. Okay, this is used to treat spinal muscular atrophy. You know what is spinal muscular atrophy? Generally, this occur only in the child, like after the birth. Okay, after 1.5 year or after 1 year, the child feel the child mental ability is going to reduce. The first year they don't find any kind of symptoms, but after first year or 1.5 year, they find some kind of mental disability. So to treat this kind of a spinal muscular atrophy, okay, so this kind of a shot, 5.5 ml, this will be given as a single shot, and the price of this single shot is 18 crore in 2018. Now I heard it's more than 25 crores, comes around 26 crores. You know, 3 shots have to be given to save a patient. 3 into 26. See the expense of treating this kind of a disease. Even the multimillionaires are affording to procure this kind of a disease for treatment. 26 crores. And you know, this is going to target one protein called SMN protein of human. For targeting this SMN protein, only Novartis has developed the medicine. And that too, how much effective it's going to work with the humans, how much precise it is. It can, according to the present statistics, it can able to treat only 1 out of 3. The remaining 2 have to die, even after taking these 3 shots. So we don't have medicine for this kind of a disease, especially for targeting the major target, human SMN protein. So like this, we have so many rare diseases. 1 in 1 million can get that. 1 in 10 million can get. So to come up with a kind of a medicine, so obviously we need this kind of computer-aided drug and vaccine design course. Okay, more about this network pharmacology and how to practice the practical approach. We'll be discussing it on tomorrow. Okay. So any doubts you can ask me at present. Do you have any doubts? If no doubts, thank you very much for your interaction. Good night to everyone. Bye.